Hello, and thank you for attending today's webinar, How Image Integrity Impacts Your Ability to Publish or Perish. I'm Mark McNulty, General Manager of Datacolor Chroma. Also joining me from Datacolor is Mark Clymer, Marketing Director for Chroma. Datacolor is pleased to be hosting today's webinar and delighted to have our guest speaker, Jerry Sedgewick, who I will introduce shortly. I do want to mention that at the conclusion of Jerry's discussion, Datacolor will announce a special offer available to today's attendees. I do believe Jerry's talk will be enough of an incentive, but I encourage you to stay for the duration in order to take advantage of this offer. So let me introduce our guest speaker, Jerry Sedgewick. Jerry has published 2 books on scientific imaging, image integrity, and image analysis. The most recent entitled Scientific Imaging with Photoshop, Methods, Measurement, and Output. He has authored and co-authored several book chapters and journal articles, including those in Science, PNAS, and Circulation, among others. Jerry ran a core microscopy facility at the University of Minnesota for 15 years, and in that time custom built a video rate multi-photon confocal. He has taught at Woods Hole and giving numerous presentations on scientific imaging and image integrity at scientific conferences. Jerry currently provides consulting services for scientific imaging and image analysis via his company, Imaging and Analysis, LLC. At this time, let me welcome Jerry, and I will turn the podium over to him.
Thank you, Mark, and welcome to everyone. So I'm going to start overall. I'm going to start with an overall view of image integrity, and then from there I'm going to go down to the nitty-gritty and actually show some images that are misleading and that could be the sorts of images that an editor would send back to you or a reviewer. First of all, I think it's useful to define image integrity, and in this instance, I consider image integrity to be a process. So it's the incorporation of a means to archive original or raw images, to record a history of changes to the image, to apply appropriate post-processing steps with the intent to better reveal obscured visual information, and not to falsify or create misleading images, and to report what's been done to images. So that's my own definition. So there's an effect to publishing false and misleading images, and the ramifications are huge. Normally in publication ethics, things like plagiarism, authorship, and those sorts of things are described and gone into in great detail, but image integrity is left sort of by the wayside. As you can see with this graph here, since the dawn of the digital age, the number of misconduct cases at the Office of Research Integrity, which is a subset of NIH, that these have increased very rapidly and dramatically, to a point at which now almost 80% of scientific misconduct cases involve the use of images. This kind of misconduct really makes the news. You know, if it's plagiarism, that just doesn't really titillate the public. But when it's something like a misleading image, that definitely gets people's attention. And the ramifications result in a loss of funding, a loss of reputation for you and the university, and of course, the thing that you don't want to have to face, which is disciplinary actions. And with the dawn of the digital age, now we have a lot more online journals, and so everyone can evaluate your research, from the general public to those who are in the scientific community. And probably as a result, it's led to now a dramatic increase in retractions from publications. And you can see here that from 2005 to 2011 that the increase in retractions has been very dramatic. Now, when talking about misleading images, I want to be really clear about the fact that, you know, there are those who intentionally falsify their images. There are those who do that intentionally, and there's not, you can do that. I mean, Photoshop's out there, you can do that. There's other ways in which you can falsify images. But in this instance, I'm talking about inadvertent misleading images, people who do this inadvertently. And this is something that's we don't often, or that too often isn't discussed, and the scientific community really hasn't caught up with editors in regard to thinking about this. Editors now hire people who are image integrity specialists, and all they do is look at images and then evaluate them. But the reason that we really need to focus on images is because images are both visual data and visual information, but really they're heavily visual data. Images are made up of pixels. Pixels have tones. The tones are given a numeric value based upon the overall number of tones, the bit depth, and then they're also spatial. You also have, as a unit of measurement, you can have pixels per micron. So in both those instances, when your image is made, it's really as much visual data as any sort of graph or measurement you might make. If you have this data, then you certainly don't want to alter it after you've acquired your image. And so in the area of spatial data, you certainly don't want to save your image in such a way that you're actually throwing away visual data. And so in this instance, you can see that the top image was saved as a TIFF image, which doesn't throw away any kind of visual data, but the image on the bottom was saved as a highly compressed JPEG. And as you can see, the data has been removed. Much of the spatial data has been removed. You might not know this, but you can also have this problem in PowerPoint, depending on the version and depending on how large your image is in file size. You'll note that in the top image that there's a far greater level of pixelation than in the bottom image. The bottom image was saved in the native format or as a TIFF from the program in which it was acquired. In terms of tones, you want to make sure that your tones also do not get altered. And in so many publications, you're looking at two conditions, one which is an experimental condition and one which is a control, or in which the experimental effect wasn't as great, and you want to say something like, this stain darker than this, or this is brighter than this. So it's important then that you keep those tones and retain them, but it's also important that those tones be linearly related. In other words, that all the tones are in a linear relationship with each other, and that relationship is preserved. So you can see on the step wedge that's on the left that you have increments of gray tones going from dark to light at the top, and on the right you can see that it's nonlinear, and now you have a lot more blacker tones toward the bottom and then the lighter tones toward the top, and you've lost that linear relationship. That relationship is assigned by gamma, and like a gamma of 1 ensures that you have a linear relationship. Any gamma that isn't 1 ensures that you have now made a nonlinear relationship. Now I'm going to pause here for a minute and ask you if you actually know whether your camera system is linear or nonlinear, and whether you have actual settings that are set up in such a way that you can be assured that you have a linear relationship of tones. I've been in lots of labs, and it seems like too often I wander in and then see that someone has changed the settings on the camera or confocal or whatever the instrument is, and all of a sudden it's on something called enhance or something called contrast, and you have no idea whether or not that's changing the gamma. And it's a surprise to me, and actually it's stunning to me that I could go into labs and see this. It's really important, at least for your acquired image, to have a linear relationship of tones, and especially when you're saying something's brighter or darker than something else. But images aren't only visual data. They also show something. They show you what it was that you once saw through the microscope. In this instance, you see that there's a Purkinje cell that's on the left, and while it looks, appropriately stained and certainly shows that staining has occurred, the image on the right looks markedly different. Now the staining is punctate or dotty. And so in this instance, the acquired image actually did not look like the image that was seen through the microscope, and so, dare I say this, and so it was sharpened. And in this instance, sharpening would be perfectly appropriate, because now you want to show that you have punctate staining. In fact, you're going to say that in your publication. And so it makes the most sense then to sharpen it and, of course, to report that you've done that. So how do you straddle that line between, okay, my image is visual data, but my image is also something that people look at, and that is a matter of perception? This is how you do it, with 4 good laboratory practices. And I call these 4 good laboratory practices RAH-RAH. RAH-RAH, record what you've done, record everything you've done to your image after you've acquired it, and record how you've acquired the image, which camera settings and equipment settings. Archive your originals, and in this instance, by archive, I don't just mean that you store it in a place, but that you also prevent people or yourself from saving over your original image, because guess what? That's what you're going to be asked for if you're challenged at any point about the images that you used in publication. And then you need to report what you've done. And I see so much hesitance on the part of authors to report that they tweaked an image or enhanced it. But these aren't necessarily, this isn't necessarily why you're changing an image. There are other reasons for doing that, and we'll talk about that a little bit more. But it's really important to report that you've done this. And then finally, if you apply post-processing, then you need to do this globally to the whole image and minimally, so that you're not going into any specific area to make a change. The big exception is gels, blots, electrophoretic samples, and any kind of image that's destined for densitometry. These images, you do not change the tones, and anymore it's a very good idea to submit the original image and not to do any kind of post-processing, except within certain limits that I'll talk about as we go on. So, how are you going to implement good laboratory practices within your lab or your work group? Well, you can do it by asking people to do it themselves, or you can use a commercial solution. And so in this instance, I have a software system comparison of a variety of different software programs, and certainly I don't have them all. This is just a scattering of programs. And in each instance, these programs can either record what you've done while you're working on the image, or they can archive it. And in this instance, I'm not talking about storage, I'm talking about ways that are instituted to prevent you from saving over the original image. Or you can, and also they would have within them some sort of way of reporting, so that it isn't up to the author to report it, but something within the image itself that actually reports to the publisher or the reviewer or whomever this is being presented to, that you have in fact done some kind of post-processing. And about the only way I can think of doing that is by putting a label on the image. And then finally, in each instance, these would apply post-processing, and all of them do that. So you can see that for Image Pro Plus, Metamorph, GIMP, and Photoshop, that they all do have a means through which you can have an audit trail and actually record what it is that you're doing, and they can all apply post-processing. Bridge and Lightroom, which are both Adobe products, and MATLAB will also prevent you from saving over the original. And there's been a fair amount of work in Photoshop to try to help that from happening, but it isn't foolproof. MATLAB, you'd have to program yourself to do that. There's a new product called ChromaCal, and I'm rather impressed with this one because it not only records what you're doing, but it also prevents you from saving over the original, and then also labels the image so that you now reported that you've done something to it, and it applies post-processing. So the way the system works is you have a calibration slide with a set of colors that you can then standardize your image to, some image calibration software so that it can so you can run through a number of images in a batch process and so on, and then monitor calibration. And I should note here that though I haven't talked about calibrating color, that's certainly an extra that's provided by this system. And I remember years ago that I wanted to do this on my own, and I'm really pretty glad now that there's a company that's actually provided this sort of a solution. So the way the ChromaCal records is it actually puts information in the metadata so that you can find it, and you can see here that the metadata was includes that the image was calibrated. There's some contrast information. It also has linearity information and so on. The way it prevents you from saving over your original is it saves a second image, and that image has the words chroma calibrated. And then it will also report by a label that you can apply to the image itself, also with information on it that lets you know what sort of corrections and dates and times and that sort of thing. And then finally, it applies some post-processing all the time, keeping the image linear. So not only does it white balance, as you can see in the first set of images to the left, but it also color standardizes and brightness matches. So in essence, what it does is it gets your image from its original state to publication and report ready. I should note here that almost every camera has a hue shift, and back in the day I was once a photographer and had always wondered why it was that in the sciences there was a sort of that this whole idea of color shift and hue shift was simply ignored. I thought, oh wait, I'm with scientists, these are people who calibrate, and in the end found out that that wasn't true. It also, there's some extras with the system, is it does check for the linearity of your camera system. And by system, I mean not just the camera itself, which very likely is linear, but with some of the settings that you might set in the camera acquisition software. And in this instance, on the software itself, you'll see that it'll show you that the linearity is poor and actually won't let you proceed. And in the graph that you see to the right, you can see that some of the color values are actually scattered above the linear lines, showing that there was a change in gamma. And then it calibrates the monitor. And you know, you have to remember that with monitor calibration, it's not just that you're calibrating colors to a standard, but you're also calibrating tones. And for those who are in fluorescence and those working with electrophoretic samples, this is really important because you do need to see further into the blacks and into the whites, and you do need to know that you're seeing everything that was on your image when you acquired it. And I've been in situations where scientists have looked at gels and because their monitor wasn't calibrated decided that they needed to brighten them, when in fact on another monitor it was perfectly bright. So the limitation of ChromaCal is that it works with transmitted color bright field images only. Now, the other thing you can do is implement good laboratory practices on your own. As you know, when you're working on your own or with groups of people, this can be a little bit of a challenge But to at least to attempt to incorporate this would be a very good idea within your work groups. You can start by using a software program that does record what you're doing in post-processing. And if you're using Photoshop, there is a function in there called history log, which will record not just to your image, but also to a text file. You can use a lab notebook and hopefully not get distracted by the many things that occur in a lab so that you don't forget to write something down. And then you can always save in the manufacturer's format, which usually in manufacturer formats, proprietary formats, there's contained within it the equipment settings and some of the exposure settings and camera settings and so on, and so that's a good idea. And then you would save a second image as a TIFF file, and that's the file you would work on. As far as archiving, you'd want to now, of course, archive to the cloud for storage, and in this instance, that would be a good idea, but you may be in a situation where you don't have internet access, and so obviously if you're going to use DVD or CD, the -R are the best to use. You'd want to store in two places, save in the manufacturer's format, mostly to prevent yourself from saving over the original. Again, you'd save a second copy as a TIFF image. And then here's my own suggestion, and that is take pictures of step wedges with gels. So why would I say that? Because if you acquire your image with a step wedge, not only are you assuring yourself that you, not only by measuring the step wedge, and these are calibrated, not only would you then assure yourself that you're using a linear system, but by having that step wedge there, you can present your original image if ever challenged, and you could prove that the readings off that step wedge match the readings that are provided by the company. And then a way that you can take steps to prevent saving over originals is use file folder protocols. And so, you know, rather than drop all of your images into one folder, you separate that out and have your images divided out as they are on the right, where you have your original images in one folder and then other images would be in a folder that you might call Photoshop corrections or image J or whatever, but certainly by dividing your original images out from your images that you're correcting. So when it comes to post-processing, there are things that you can do to the image. You can change, and again, when I'm talking about the image here, what I mean is a representative image, an image that's a representation of what you once saw through the microscope. And I divide this out from images that are intended for densitometry and a third kind of image that would be intended for measurement, and so, or for morphometry specifically. What you can do to the image is change the whole image globally and not go in and tweak the channels or go into specific areas. And when I talk about channels, what I mean is that it is perfectly appropriate from the red to the green to the blue channel to adjust them to fill the dynamic range, which is called normalizing. That's an appropriate and community accepted standard. But it certainly isn't appropriate to go into any of those channels and make them look brighter than the other two arbitrarily. You would want to apply the same changes that you did to the whole image to the controls. Again, you'd want to minimally process, and then finally you'd want to report any gamma changes that you might have made, because there are times when gamma changes are perfectly appropriate. If you want to reveal obscure detail, you may have to change the gamma in order to show that. So just report it when you do that, and certainly you wouldn't want to measure from an image in which you've done that sort of gamma change for optical density or intensity. And then the editors say this to me now and again, that they get these images in which people just decided that they didn't have enough cells in one image, and so they decided to borrow from other images and drop those in. No, you're not supposed to do that. As far as gels and blots and images that are destined for densitometry, you know, you can crop those, and again, you'd want to look at the author's guidelines to see how they want those cropped. You can resize them, letter and label them, And it's very important if you're not using a scanner and you're using more of a camera system that you correct for uneven illumination, and I'll talk a little bit about that. And then it's important if you're doing a longitudinal study where you may have differences in light levels, it's perfectly appropriate to do a linear histogram shift or as a kind of brightness matching. And another area of images, or a third area, are those images that are really meant for measurement of features such as counts, areas, lengths, widths, which is generally called morphometry. And if you're using images for visualization, and that would include 3D reconstructions and cartoons and pseudo-coloring, then you would expect that filtering would be done, and you would expect extensive post-processing. It takes quite a bit to actually divide out, as in the images below, where you see on the image on the left the objects of interest, which happen to be the stained small areas, and then the image on the right is the one that you would actually measure, and that takes a fair amount of post-processing. Again, you'd report all of that in your methods. So let's now go on to the sorts of images that are misleading and the kinds of images that we would anticipate a reviewer, an editor, or someone who's actually looking at your work to contest. So first, is your signal real? In this instance, you can see that the signal is very close to the background. In this instance, you could increase your contrast and exposure, but the difference between the background and your image itself, and here I drew a line and made a line profile, and you can see by the arrows that there isn't much of an increase from the background to the actual fluorescence. And so in this instance, you would really have to go back and review your bench techniques and figure out how you can improve your signal to background problem. This one is really actually way too typical. Clean backgrounds. And by this what I mean is that you've made the background so white that it's like, wait a second, what are you hiding? Is there a faint band in there that you don't want me to see? Or they make it so black that you can't see any background detail, and this happens way too often in fluorescence. And furthermore, it could be that the backgrounds are clean because somebody's gone in and kind of, touched up all the non-specific staining, so-called non-specific staining, and sometimes it is non-specific, but it's not appropriate to remove it. This one is probably the number one problem with images, and I see it so often, it literally it drives me nuts. Why would you acquire an image and then literally remove the details in the brightest part of the image? You could see the image on the left that you can see all of the detail in this pollen sample, but on the image on the right, now some of the detail is blocked and you have saturated that part of the signal. You always acquire your image so that you can see all of the significant areas of the brightest parts of your image, and immediately when you see things brighten like this, you think to yourself, oh yeah, they want me to think that they got lots of staining. But this is not appropriate, absolutely not appropriate. And then if there is issues with uneven illumination, and you'd have to look at this image a little bit carefully, but you can see that there are 6 spots which actually are all of equal brightness. And then you can see in the background that it's kind of a little less dark at the edges and a little darker in the center. And if you draw a line profile across that, you could potentially measure this incorrectly because you would think, oh, I should measure along the baseline. And if you measure along the baseline, you're going to end up getting erroneous readings. And so I'm going to say to myself, oh, can I trust their measurements? Unless I see somewhere in the methods that they've done some sort of correction for uneven illumination, which is normally a method called flat field correction. And when people over-sharpen an image or they don't set color illumination correctly, you can end up with information that simply isn't there in the original. And so if I were to see the image that's on the right, I just think to myself, hey, what is going on here? You know, this thing looks like it has the measles. And so, you know, the thing is that this was taken incorrectly and or over-sharpened. Well, in review, you can see that image integrity really matters. It makes the news. It gets you right on the front pages of the Huffington Post. So, you know, you really need to think about the decisions you're making. You really need to think about what it is you're doing and the consequences of those decisions. And if it turns out that you are inadvertently pegged for scientific misconduct or for a misleading image, or your image is sent back, and especially if it gets worse and it becomes something that's publicized, you know, that will result in a loss of funding, loss of reputation, and disciplinary action. And images are visual data, and you'll hear this a lot, but they're also visual information. They're also ways in which you are showing obscured or hidden details that should be revealed in order for those who are reading your paper or looking at your report, in order for them to see what it is that you had seen yourself through the microscope. You want to keep your tones linear, and you want to make sure that your camera system is producing linear images. You want to save your images properly. In other words, you don't want to throw away spatial information by saving it as a highly compressed JPEG, and it's not appropriate to save in ways that you would sacrifice any of that kind of information. And then you'd want to incorporate the 4 good laboratory practices, and I call those rah-rah, and we went through that pretty extensively. And if you want a color bright field solution for all good laboratory practices, you'd really want to look into ChromaCal. I'm using it right now to do a paper in which I am using color as a means to segment images, and the beauty of this is I can set only one threshold point and work with the images. And then you want to think about your post-processing in terms of the kind of image that you're looking at. You can implement these do-it-yourself practices, and then you, in your lab or in your work group, and I would strongly encourage you to do that. And then there are those images that you would consider misleading, and those images would be those with low signal, like we saw with the GFP image, with a clean background. And again, you want to show some detail or some gray in the white parts of your images when you're working with bright field, and when you're working with fluorescence, you want to show some background detail. And then stay away from saturated pixels. Make sure that when you acquire your image, you haven't saturated any of your pixels, or at least the ones, there's obviously non-specific information that could be saturated, but anything that's significant. And then if you're doing measurement densitometry, you certainly want to avoid uneven illumination, but I should add that uneven illumination is presenting your image in a way in which it's difficult to assess, and for that reason it's to be avoided. And it's not, and so you need to think about that as well. And then avoid over-sharpening images, and learn how to use a microscope, especially when it comes to color bright field or bright field imaging, because the way in which you set the condenser aperture is critical, and that's called Kohler illumination. And before I forget, I will also let you know that when you report, if you are expanding the dynamic range of the image to fit within the dynamic range, that's called normalization. It's not called enhancement. And if you are revealing details that are obscured, then that's revealing details that are obscured. And so when you make a post-processing correction and you have a good rationale, then make sure you say that when you report what you've done. Thank you, and I'll hand this over to Mark.
Thank you, Jerry. As always, Jerry provides an outstanding overview on various topics in image integrity, and certainly this was no exception. But before we move on to the question and answer portion of the webinar, let me first mention a offer available from Datacolor for attending today's webinar. For a limited time, details are on your screen. The first 25 attendees who order the ChromaCal system will receive the calibration slide free. This is normally a $650 value. And all you need to do is provide that promotion code, free slide 123 at time of ordering. So ChromaCal is available through Electron Microscopy Sciences. Their number is shown on the screen. And certainly we encourage you to take advantage of this promotional offer. At this point, I'll turn it over to my colleague, Mark Clymer, to lead us through the question and answer session.
All right, well, thank you, Mark. If there are any questions, if you would mind entering them into the question portion of your control box, that would be great. We did receive one question that is I guess a little technical into the colorimetry space, and that's something I think we will discuss, I think, offline, but we do have a response from our optical engineer on that. Another question, can splicing together bands from separate gels ever be appropriate? And let me unmute Jerry and let him provide that response. Jerry?
Yeah, according to Nature in their author's guidelines, you can, if you put lines between them, you can do that. You just have to make it clear. And again, you know, that's Nature's author's guidelines. You know, each journal will have its own guidelines, but yes, it's appropriate if you make it really evident that you've taken your bands from different gels.
Okay, thank you. Are there any other questions? Please use that enter the question section of your panel. Any other questions? Here we go. As to JPEG compression, any comment on a commercial image analysis expert who used such a JPEG image instead of the original TIFF image and then concluded that the image was falsified?
That's a good question. Yes, when it comes to, from my understanding, and my understanding is limited, but from my understanding, the image forensic programs that look at whether or not an image has been manipulated, my understanding is that it's a little bit more difficult with JPEG images. But honestly, I don't know enough about that particular area to comment any further.
Well, Jerry, let me ask a follow-up question myself. Since editors and reviewers request usually the images in the form of JPEGs, considering online publication, how do you reconcile them between the JPEG and the TIFF and the compression artifacts?
There are different programs that provide, well, Photoshop, for an example, provides varying levels of compression. And if you choose a compression level that's in Photoshop what they consider maximum, which is a setting of 12, actually the difference between your image and the compressed image is virtually undetectable by eye. So really it depends a lot on how compressed the JPEG file is.
Okay. I have another question here. Does ChromaCal compensate for changes in light sources? For example, bulb age, and how does ChromaCal work with proprietary platforms and image formats? Are there any tools for error checking images to look for irregular or questionable features, such as excessive Photoshop use? I'll take that one. ChromaCal compensates for a variety of light source effects by the nature of the way of the workflow. So you would capture an image of the calibration slide, which Jerry showed earlier in the presentation, and that image becomes the color fingerprint of your imaging session. So therefore, it's going to use that then to determine a correction matrix to be applied to your images. So, and it calibrates the images during the same session. So thereby it will account for changes in light sources. As far as proprietary formats, ChromaCal is only working with TIFF and JPEG images. At this point, we are not opening proprietary formats, ScanScope, Apereo, Zeiss, Nikon. They should all be exported as TIFF or JPEG, and be careful not to export with and applying any of the display properties, that sometimes will apply some gamma to your image. And are there any tools, imagine within ChromaCal to check, error check for irregularities? Nothing that we offer. ChromaCal is only looking at strictly at color on a pixel-by-pixel basis. It's really quite ignorant to the acquisition source and really what the image is altogether. And I don't know if there's anything commercially available that you can use to explore your images to see if they've been photoshopped, for example. Jerry, do you know of any?
Yeah, actually the editors are using, there's 2 or 3 image forensics programs, and there are some functions you can do in Photoshop to detect it, but generally these are, generally from editors I've talked to, they find that humans are still the best source for figuring this out.
Okay, Jerry, how many, may come back to you, how many people actually get caught and in trouble with regard to misleading images?
Yeah, honestly, I don't have a number. I just have the graph from John Kruger at the Office of Research Integrity that he simply started to track it because he was seeing a trend.
Yeah, I don't know that NIH is actually scrutinizing. It may be reported to them possibly by reviewers or editors.
Correct, that's what's happening.
Right, and then they would proceed to investigate from that point. But as Jerry mentioned, there are image forensics capabilities out there that do explore questionable images on behalf of publishers. We have a question that the asker feels that the button like brighter on the Leica confocal may apply some enhancements like undeclared gamma to the image, and should probably be blocked. We believe that this is a situation not just on a confocal, but we see this in many color cameras with bare mask filters, where the intention is really to provide an image that is comfortable and is applying some gamma, as our eyes naturally do, as we view an image through the microscope. But it would be nice if it were a little more apparent and not as hidden, and maybe some consistency within the camera scientific camera industry, so we know what buttons to turn on, what buttons to turn off for those purposes. So we can maintain linearity in our image. You can always apply it later, after the fact to improve visualization. Are there any different? Let's see. That was a color imagery question. Is there any way to tell if an image has been sharpened, lightened, or otherwise modified by looking at a histogram? Jerry.
Generally, when you change tones, you can look at the histogram, and what you'll see is spaces between pixels. And so generally, I would say the answer is yes. Now, there are ways around that. Actually, you could make your changes, and then you could put in a slight blur, and that would reform pixels at certain locations. But in general, yes, you can tell when an image has been post-processed, because usually, as a result of rounding errors, you end up with positions in which there are no pixels. So you can see that on the histogram.
Okay. And there was a follow-up question asking if there's any way to look at images forensically to determine if it's been modified, and the answer to that is yes. There are companies that do that. I don't know exactly what they're doing, but
also, if you go to the Office of Research Integrity, ORI, they actually have like about 10 things you can do in Photoshop to actually determine whether or not an image has been manipulated. And I would encourage you to go there to look at what they're doing, and then you can apply that yourself in Photoshop.
Let's see. As far as reporting any changes, I believe there was another follow-up to this. I'm not sure there's a particular format in which to follow, but guidelines when you do submit your images with a manuscript for publication, that there's a suggestion of what you are to declare. Usually it's included within your methods section. We have a question here about suggestions on antibody validation. I'm not really clear how we can answer that without a little more information as to what you're trying to achieve with regard to antibody validation, except maintaining perhaps and looking at controls and maintaining a good histogram on a negative control, making sure that your histograms are similar between the two so that you can differentiate between a positive response with an antibody and a negative.
You would also want to look at lasers at different channels to make sure that your staining is specific and not non-specific.
Correct. Right. Are Photoshop autocolor or auto contrast tools and similar automatic adjustments in other programs appropriate form of image processing?
That's the million-dollar question. If I am hesitant to use the auto color or auto contrast because I don't know what it's doing. In Photoshop, there is, if you go into levels, you can actually open up a window to tell Photoshop how many pixels you wish to have saturated. So the real issue comes with when you use auto contrast or auto color, how much of your image is actually getting saturated and where are you losing detail. And because of those functions, especially in programs like Photoshop, because those functions are adapted for pictures of mom and dad and grandma and grandpa, you know, you have no idea how many pixels they're deciding to saturate. So at least in levels or when you have control over how many pixels you want to allow to have saturated, and they have this in programs like ImageJ, then when you use those functions, that they can be appropriate as long as you also check to make sure that they keep your image linear when linearity is important when you're describing that something's brighter or darker than something else.
Okay. There's one other question here. Are there any plans to add calibration tool for fluorescence? Currently, Chromacal is available for bright field, and I should say transmitted bright field. It's not suitable for reflected light applications. Fluorescence is an interesting area and something that we do plan to explore in the future, but we do not have anything at this point and no definite timeline. But we would like to contact us separately. We would like to have some discussions about what is needed in the area of fluorescence calibration. And let's see, oh, here's a comment back. Use of appropriate assay controls can eliminate the need for image editing. I think this is related to the antibody question.
Yeah, that's a little too vague. I'm sorry. I don't quite know what that means.
Just controlling, have your proper controls when you're setting up your assays, the dilutions and so forth, the non-specific binding. Okay. If there are, I don't see any other new questions at this point. So if that is all, what I would like to do then is thank everyone for your attention during the webinar. There are a number of outstanding resources available on the internet. We do make them available here to you and through our website, chromacal.com. Also, you are welcome to contact us directly at our emails that are towards the bottom of this slide. If you have any questions or would like some additional information, I will be happy to reply to your email and provide that information. I would also like to inform you before we close the webinar that we will be making a recording of the webinar available in the near future. It will be available on our website. We'll send an email to all of the registrants of this webinar as to when that is available and provide a link for you for your convenience. You are welcome then to go in and view the webinar, also to share the link with any of your colleagues who missed it and would like to view it separately. And just check one more time for any questions. And I do not see any questions. I believe that is it. I thank you all for your attention, and please have a great day, and always think of integrity when you're imaging. Thank you very much.